In today's video, we're going to be talking about neurons, what they are and how they work. Neurons are the cells that your body uses to send messages. They regulate emotions, contract muscles, and provide your brain with information about the environment. They're kind of like cell towers that allow you to send text across the body. There are three main components of a neuron: the cell body, the axon, and the axon terminal. The cell body contains the normal organelles found in all cells. The difference is that it branches out into dendrites. The word dendrite actually comes from Greek and means of or pertaining to a tree. Dendrites receive signals from other neurons. Once a signal is received, it's sent down the axon. At the end of an axon are axon terminals, where the signal is then passed on to neighboring neurons. An axon can be myelinated or unmyelinated. Myelinated axons are wrapped in Schwann cells, otherwise known as myelin sheaths. The space between these sheaths are called nodes of Ranvier. As you can see from this lovely animation, on the right side we have a myelinated neuron. The electrical signal seems to be jumping in between these nodes of Ranvier down to the axon terminal. Signals can be sent much faster in myelinated neurons compared to unmyelinated. Next, we'll talk about how a signal is sent through a neuron. This involves what's called an action potential. This action potential is possible because of a difference of electrical charge between the inside and the outside of a cell. Here is a diagram of a generic action potential. There are four distinct phases. A neuron starts at its resting potential. If it receives stimulus that pushes the potential above a threshold, the neuron fires. It becomes depolarized, then peaks, and repolarizes. It overshoots the resting potential and becomes hyperpolarized. Finally, we return to its resting potential. Polarization refers to the separation of positive and negative charges from inside and outside the cell. This is another diagram of an action potential, but now we have membrane potential on the y-axis. Alright, so membrane potential gives us a measure of the difference of charge between the inside and the outside of the cell. Here we can see that the inside of the cell has a charge of roughly 70 millivolts less than the outside. Negative 70 millivolts tends to be the average resting potential of neurons. Now, if we come back to this diagram with membrane potential on the y-axis, we can see that the resting potential is at negative 70 millivolts. We have a dotted line at negative 55 millivolts, which is the threshold before a neuron fires. Neuron firing is considered all or nothing. So if the stimulus pushes the potential above this dotted line, it fires and becomes depolarized, reaching plus 30 millivolts. The process of moving an action potential is controlled by sodium and potassium ion channels. These channels are in the cell membrane and allow the transfer of sodium and potassium between the outside and the inside of an axon. A trick I learned from Bozeman Science is to remember neurons like salty bananas. Sodium ions are abundant outside the neuron, and potassium is abundant inside the neuron. When the action potential threshold is broken, sodium ion channels are opened. Sodium rushes in, depolarizing the neuron. Sodium channels close, and then potassium ion channels open, allowing potassium to rush out. This causes the repolarization of the neuron. After overshooting repolarization, the potassium ion channel closes. A sodium-potassium ion pump begins working to stabilize the neuron at resting potential. It does this by pumping in 2 potassium ions and pumping out 3 sodium ions with the help of ATP. Here we have a transverse slice of a neuron, and I'm going to use this to show you a simulation of how a neuron fires. If we zoom in a little bit, we can see the sodium and potassium ion channels located in the membrane. And then if we stimulate the neuron, we can see the action potential come in, the sodium rushes inside, and then the potassium rushes out. It takes fractions of a second for a neuron to send a signal down to the axon terminal. If you want to check out this simulation, you can find it at phet.colorado.edu. I'll link this website in the description. You can find this simulation along with many others. Another thing I wanted to mention is that the intensity of a signal is not determined by how much the neuron is depolarized. It's determined by the frequency of fire. The faster a neuron is firing, the more intense the response. I chose this picture to illustrate how the brain codes for signal intensity because it shows another phenomenon known as adaptation. A stimulus is applied, and the neuron frequency is high. Over time, the rate of fire slows down. You can test this yourself if you pinch yourself. You'll notice a sharp pain, but as you hold, the pain slowly decreases. This is because our senses evolved to sense change. If a stimulus is constant, our brain will drown it out in order to be able to pick up more subtle changes in our environment. Now we're going to talk about what happens at the ends of a neuron. This is where the signal is passed on to the next cell. The point where one neuron ends and the next begins is called a synapse. There are two main types of synapses, an electrical synapse and a chemical synapse. Electrical synapses physically connect to perform their function. They use what's called a gap junction to pass information along. These synapses are generally used in muscle tissue and in some brain regions to allow for synchronized action. Chemical synapses are going to be the focus for the rest of this video. So before we go further, I want to make a distinction between the two neurons involved in signal transduction. There is a presynaptic neuron, which is the one that's sending the message, and a postsynaptic neuron, the one that's receiving the message. Chemical synapses convert the electrical signal into a chemical one. They do this with the help of mitochondria. Some neurons are estimated to have hundreds of thousands, if not a couple million mitochondria at their disposal. Neurotransmitters are stored in vesicles located at axon terminals. These neurotransmitters include dopamine, GABA, acetylcholine, and many more. Autoreceptors, or self-receptors, are able to detect the neurotransmitter released and help regulate how much is being exposed. This gap between the two neurons is called the synaptic cleft. On the postsynaptic neuron are G protein coupled receptors that bind specific neurotransmitters. Up at number 6 are calcium ion channels. Calcium triggers the release of vesicles. Once the electrical signal triggers the release of neurotransmitters, it travels across the synaptic cleft, carrying information about the original stimulus. And finally, we have a reuptake channel that allows the neuron to reuse the discharged neurotransmitters. As I said earlier, neuron firing is considered all or nothing. In order for a neuron to fire, a threshold must be broken. If this threshold is not broken, then the neuron will not fire. Oftentimes, a neuron will have to add together potentials received from a presynaptic neuron. This is called summation. Summation can be done temporally, meaning over time, or spatially, from two or more presynaptic neurons inputting at different areas of the cell body. In Figure A, the action potential received is below the threshold, thus no firing occurs. In Figure B, the postsynaptic neuron receives two bursts of potential from an excitatory neuron and adds them together to break threshold. The neuron fires. In Figure C, a postsynaptic neuron receives a burst from two separate axon terminals connected at different locations and adds them together to break threshold. The neuron fires. In Figure D, the postsynaptic neuron receives an inhibitory impulse and an excitatory impulse that sum to a potential below the threshold, so the neuron doesn't fire. I'm going to use a dopaminergic neuron as an example of a neuron that uses a chemical to pass along a signal. Dopaminergic means that the neuron uses dopamine as a stimulus. This image appears more complicated than it is. Many of the stuff we've already talked about, so we're going to break it down piece by piece. Down at the bottom in the synaptic cleft is the released neurotransmitter dopamine. These things labeled D1 through 5 are dopamine receptors. These are GPCRs. I've made a video discussing G protein coupled receptors that you can find on my channel already. This one labeled DAT is the reuptake channel. Its full name is dopamine transporter and pumps released dopamine back into the neuron. COMT and MAO are enzymes that break down dopamine. HVA and 3-MT are metabolites of dopamine. These are the pieces that the enzymes broke dopamine into. The body usually gets rid of neurotransmitters in the synaptic cleft either by reuptake or breaking them down. Sometimes neurotransmitters can diffuse away and become too dilute to have an effect. Here we have a dopamine vesicle where the neurotransmitter is stored. And the pathway leading into the vesicle is how dopamine is made inside the brain. We start with the amino acid tyrosine. An enzyme abbreviated as TH converts tyrosine into L-dopa. The enzyme DDC converts L-dopa into dopamine. VMAT1 and 2 signifies transporters that move dopamine into the vesicle for later use. As I was writing the script for this section of the video, I found out that the gene for VMAT is what has been dubbed the God gene. Dr. Dean Hamer suggests that humans are more inclined to be spiritual depending on the expression of the VMAT gene. From what I read, I think the media just picked this one up and ran with it. Anyways, to put it all together, a dopaminergic neuron is stimulated, an action potential is sent down the axon, and dopamine is released from the axon terminal. The postsynaptic neuron detects this signal using its dopamine receptors, and the signal continues along the chain. If you would like to know more about how dopamine works and how its release is regulated, I recommend checking out this article published in 2019. I'll make sure to have the link in the description below. To wrap up this video, I'll introduce you to the neuroanatomy that you can see above the neuron. Neuro means relating to the brain, and anatomy is relating to structure. Dopaminergic neurons are found in abundance in the substantia nigra, which is this dark pink strip located in the midbrain. The substantia nigra is involved in the reward response, which is why some call dopamine the happy hormone. The substantia nigra is also involved in motor movement. Defects in dopaminergic neurons leads to symptoms of Parkinson's, specifically the shakiness often associated with the disease. Dopaminergic neurons create the nigrostriatal pathway that connects the substantia nigra to the striatum in the basal ganglia. The two pieces of the striatum being the caudate nucleus and the putamen. Like the substantia nigra, the striatum plays a critical role in reward response and motor function. Thank you all for watching, and I hope you learned something new.